Meiosis is incredibly important to understand because it is how life generates genetic diversity. How does life adapt to new situations and become more fit to an environment? And meiosis is the key to that. So if you were to summarize, what makes up the heart of biology, it is meiosis. And meiosis, as we know, is a double-edged sword. In some cases, it gets us genes that are that give us phenotype advantages in new environments, and in other times it can give us things that hinder our performance, and that's when we get genetic diseases. But it is a very important concept to understand, and so what I'm going to do in this video is go over an overview of meiosis, and the overall thing that's happening here is we're going to take a diploid, referred to as a gametocyte, and we're going to generate 4 unique gametes. And these gametes are haploids, meaning they just have one chromosome, and that differs from their parent, the gametocyte, in that it is a diploid. And so these diploids will have 2 copies of each chromosome. And so the key takeaway between meiosis and mitosis happens in this meiosis 1 phase. And in this meiosis 1 phase, the first thing that we're going to see is exactly what we've seen in mitosis, but with some very important additions. And the first addition is something referred to as synapses. And the big thing with synapses is we're going to have crossing over in tetrads. And so just like in mitosis, with prophase, in meiosis 1, in prophase 1, the nuclear envelope will also disappear. The centrioles will move to the 2 poles in the system, and the chromatin will condense into chromosomes that we can actually observe much more easily. But in addition to that, these 2 chromosomes will form tetrads. And these tetrads are physically linked together, and the genes will break off and reassemble in such a way that we are generating genetic diversity. So in many ways, prophase 1 is at the epicenter of the heart of the evolution in life. And so prophase 1 is incredibly important as well in the big picture. And another note here with prophase 1 is we see, or we can guess, the proximity of genes relative to each other based on how commonly or uncommonly we find them in the gametes later on. And so if 2 genes are really close together during this crossing over stage in synapses in prophase 1, it's going to be pretty unlikely that they're going to break off and be separated. Whereas if you've got 2 genes at opposite ends of this chromosome, so like if you've got a gene there and a gene there, it is very likely that when crossing over occurs, those genes will be separated. And so that allows us to generate these kind of gene maps on chromosomes that can be useful. And a final note with prophase 1 is these centrioles will begin to attach to the kinetochores on these tetrads. And the important distinction here between prophase 1 of meiosis 1 and mitosis is that only one kinetochore, actually only one microtubule attaches to one chromosome. And this is in prophase 1 only. And this is because this is how this diploid parent, the gametocyte, will turn into a haploid daughter. And so prophase 1 is kind of the bulk with all the key distinctions that you really need to remember in a biology context. And so prophase 1 is the biggest thing to know in meiosis. If we now move on to metaphase 1, what you'll note is how, like in mitosis, we see the chromosomes, in this case, the homologs, are lining up along the middle of the cell. And also note how we only see one microtubule per chromosome in this case, and that's because that's how we're going to be doing this in meiosis 1 only. And after these kinetic cores attach, these spindle apparatuses will form other microtubules with each other, and that's also occurring during this phase. And that's what's causing this disjunction to happen at this point here, when we're actually pulling apart the homologous pairs into individual chromosomes that are going to themselves make up 2 daughter cells. And so as I was alluding to, what's going to happen next in anaphase 1, as what happens in anaphase and mitosis, is these microtubules will continue to extend as they were in metaphase 1, and these will push each other apart and also pull the homologous pairs apart. And another thing here is we're looking at a very simplified cell here with only one pair of homologs. In human cells, we would have 23 pairs of these, and so the pictures will be quite a bit larger and a little more difficult to follow, but this is just to convey the general principles. And so anaphase 1, like in anaphase and mitosis, is very similar. And then finally, in telophase 1, what we see happening is the nuclear envelopes may begin to redevelop in the 2 daughter cells. We will also see the chromosomes decondense back into chromatin. And if you're curious, the reason why it needs to decondense is if it enters some kind of interphase in which between meiosis 1 and meiosis 2, these cells need to be metabolically active in any capacity. They're going to make proteins and translate these genes. You can't access these genes like DNA polymerase, mRNA, or RNA polymerase can't access genes on these chromosomes because the DNA is so compact when it's in a chromosomal form. But when it's in chromatin, it is much more easily accessible and able to be translated and expressed as proteins. And so a key point here is after we see the cytokinesis occur, in which the we have this cleavage appearing and the 2 daughter cells are pinched apart, we now end up with 2 daughters that are now haploids. So they only have 1 chromosome each. They don't have a pair anymore. There's no more homologous pairs. And the key distinction here is, now note how we've got 2 microtubules per chromosome. And this is a key distinction because now at this point we're actually going to be separating the sister chromatids apart. And that's how you're going to go from 1 cell to 2 cells to 4 cells via this process. And if you're familiar with mitosis, meiosis 2 is very, very similar because we're not having any crossing over. There's no synapses, and it's much more straightforward. In anaphase 2, we see again what we expect after the After my metaphase 2, these microtubules also will be connected, and they'll push off each other to push and pull the sister chromatids apart. An important note here is that each one of these cells are copies. So these are identical copies. Or they can be, depending on how the crossing over occurred in meiosis 1. And then after that, we enter into telophase 2, in which we see the nuclear envelopes reforming in our daughter cells, as well as we may begin to see we will see the chromosomes decondense, again for the same reason, so that the genes can be accessed, and we will also see a cytokinesis in which the cytoplasm is pinched off to form the two daughter cells. And so this wraps up the overview of meiosis and how it relates to mitosis, and the key things to take away from it. Again, prophase 1 is the highlight, the epicenter of what you really need to take away, because that's where we are generating the genetic diversity that is a requirement of life, so that it can adapt to new environments. And so I hope you guys find this useful. Let me know if you have any questions, and thanks for watching.